next speaker is Patrick Fallet. Patrick joins us from the University of Neuchâtel in Switzerland, where he is currently a PhD student. He obtained his master's degree at Neuchâtel as well. His broad area of research is biological control, chemical ecology, and plant defense. And his research on fall armyworm in particular is biological control and chemical ecology, and especially biological control involving nematodes, which is what he's will be talking to us about today. He indicates that he has supervised MSc students and also bachelor's degree student, and he gives practicals in ecology and chemical ecology. El ponente que sigue es Patrick Fallet. Patrick nos acompaña de la Universidad de Neuchâtel en Suiza, donde ahora es estudiante de doctorado. En esa misma universidad obtuvo su maestría en ciencias. Su área de interés es control biológico, ecología química y defensas vegetales. Y estas mismas áreas de especialización o de interés las ha aplicado en el caso de gusano cogollero, con énfasis en el tema que nos va a platicar hoy acerca de control biológico de Spodoptera utilizando nematodos. Aún siendo estudiante, ha podido supervisar a 4 estudiantes de maestría en ciencias y otro estudiante más de bachillerato. Entonces también da cursos prácticos en ecología y química, ecología química, perdón. Entonces, sin más interrupción, le paso el micrófono a Patrick. Patrick, without further interruption, I pass the microphone to you. Thank you very much for being with us today. And so get started when you can. Please share your screen.
Yes. So thanks for the introduction. Can you see my screen now? Or
yes, we can. Can you
yes,
try?
Okay, very good.
Yeah.
So yeah, thanks again for the introduction. And it's a big pleasure for me to be here with you today and to talk about the project. I just recovered from a little cold, so I hope I'm not gonna cough too much during the presentation nor lose my voice. And if this happens, very sorry already, but let's hope it's gonna be all fine. So today, as Julio mentioned, I'm going to talk about the step that we took to achieve or to go towards an achievement of biological control of the fall armyworm with native entomopathogenic nematodes. And this is a project that we have in collaboration with KB and with Rwanda Agriculture Board. And this project is supervised by Stefan Töpfer from KB and by Ted Turnings from the University of Neuchâtel. So first, what are entomopathogenic nematodes? So they are very tiny soil-dwelling roundworms that are about 1 millimeter in length, and they are present in soil worldwide, so they can be isolated everywhere in the world. And it is important not to confuse entomopathogenic nematode, which means insect-killing nematodes that are beneficial organisms, with plant parasitic nematode, which are pests. So it's 2 completely separated organisms, and entomopathogenic nematode cannot infect your plants. In the soil, nematodes can locate and then penetrate into insects, and then once they are inside, they will release a bacteria that they carry with them. So it's a symbiotic bacteria, and the bacteria will in turn release toxins that will cause the death of the insect, which generally occurs within 24 to 48 hours. And then the bacteria will digest the insect tissue, and the nematode will feed on the bacteria and at the same time go through several cycles of reproduction until the food is completely depleted. And at this time, the nematode will leave the cadaver. This is what we can see here. Don't know if can you see my mouse? I hope so. And so the nematode will leave the insects, and that's what we can see on this picture. Thousands of little nematodes coming out of one insect. And then they are back into the soil and they can look for a new host. So the good thing with this nematode is that they can be mass produced rather easily. So there is mass production facility around the world, also in Africa. And they are safe for farmers because they cannot infect humans, and they also cannot infect animals, and they are considered safe for the environment. Nematodes are the third most traded biocontrol agents after Bt bacteria and Trichoderma wasps. They are commonly used against soil-dwelling pests because they are soil organisms, but not so often against above-ground pests because the nematodes are quite sensitive to abiotic factors such as desiccation, UV radiation, and temperature, which are all factors that obviously occur above ground. So now you may wonder why we would like to use entomopathogenic nematode to control the fall armyworm, which is definitely a leaf pest. This is because actually the maize fall armyworm system is quite well suited for the nematode because, as you know, the caterpillars are mostly feeding deep into the broad leaves of the world, as you can see here. And at this, on this spot, the leaves will actually provide a natural protection to the nematodes. So there will be more humidity down the well, there will be less UV radiations, and there will be also less heat. And then once you apply the nematodes into the well, the nematode will be in the close vicinity to the caterpillars, and we hope that they will be able to actively locate and move towards the insects and infect it and kill it. So the step that we took to go towards an achievement of biocontrol of the fall armyworm with native entomopathogenic nematodes are first to isolate native nematodes. Then we looked at which species should be used against the fall armyworm. Then how to apply the nematodes above ground. And then we tested our approach in laboratory trials. And then we did a first set of field trials where we only applied our treatment once. So that is a proof of principle. And then this is still a proof of principle, but we did a second batch of field trials where we had repeated application of our treatments over a whole growing season of maize. So why do we want to use native entomopathogenic nematodes? So the reason for this is that we do not want to introduce foreign species in the target country. So this is to avoid all the consequences that introducing new foreign species can have on the ecosystem. And this is also because there is a lot of barriers when you want to introduce a species. So by using native nematode, you avoid the legislation hurdles that are associated. So to isolate nematodes, we use conventional soil beating techniques, which is actually rather simple. So first you collect soil in the environment, and then you put some insect into your soil that will act as a bait for the nematode. And after a couple of days, you can collect the insect cadavers that are likely killed by nematodes. And then you can place them in something called the white trap, which is actually a technique to harvest nematode. So this is what we see on the bottom right. And with a little bit of luck, you put your cadaver in there and then you will isolate some nematodes. So we did that with a team of Rwandan scientists in 2018 in the southern and western province of Rwanda. And we took 208 cell samples, out of which only 2 yielded nematodes. So we had a success of 1%. So don't be too pessimistic about this. In general, results are more around like 10%, 15% positive sample when people do surveys of nematodes. But one of the reasons that could explain why we were like so little successful, let's say, it's because we mostly targeted agricultural fields because we wanted nematodes to be already sort of adapted to the field conditions. But as you know, agricultural fields are very much disturbed. So it may not be the best environment where you can find nematodes because there is too much disturbance and then potentially the nematode die in such soils. So as we found 2 nematodes, but the same team 2 years before our survey, they did another survey in the northern province and also in the western province, and they found 4 nematodes. So in total, we had 6 nematodes from Rwanda. Then we identified the species of this nematode using sequencing techniques. And so we extracted DNA, then we sequenced specific genes. Then we compared these genes to a gene bank, and then we managed to identify the species and build phylogenetic trees. The problem with this technique is that several species of nematodes are very closely related to one another. And also there is a lot of species that are not yet described. So this technique may lead to misidentifying closely related nematode species. So for example, 3 of the 6 nematodes that we isolated, using this technique, the results indicated that they were Heterorhabditis bacteriophora, which is one species of nematode. But actually they were like forming in the 3 a little separation. So we decided to collaborate with Dr. Ricardo Machado from the University of Bern, which is a specialist on nematode taxonomy. And he brought our investigation further, where he did some morphological characterization of the nematodes, and he also did some cross-hybridization experiments. And for these 3 nematodes, that's the sequencing indicated that they were Heterorhabditis bacteriophora. He actually found out that they were a new species that he called Heterorhabditis rwandica. So this further indicate that it is safer to use native nematodes to avoid introducing foreign species in a target country. So out of our 6 strains, 3 were Heterorhabditis rwandica, 2 were Sternernema africanum, another new species, and 1 strain was Sternernema carpocapsae. This is already described. So now the question is, which of these nematodes should be used? So we know from the literature that most nematode species are pathogenic to fall armyworm. But what we don't know is that are locally isolated nematode as pathogenic to fall as commercial nematodes? So can we use locally isolated nematode, or should we use commercially available nematodes that are often selected for their high virulency? Then we also don't know if nematodes isolated from the Americas, so from the area of origin, would be more pathogenic or maybe less pathogenic to fall armyworm than nematodes that were isolated in another continent, so in the invasive range of fall. So is there any coevolution between nematodes and the fall armyworm, and is it beneficial or not? And finally, can we see whether there is a species that is particularly better than the other? And is there a species that we could recommend? So to do this, we did a very large screening with the master students, Lara Dejani, where we had nematodes from Rwanda, from Mexico, and from commercial sources. In total, we had 40 strains representing 12 species. And we infested fall armyworm with all of these nematodes. And after several rounds of first screening, we identified the most virulent nematode from Rwanda, the most virulent from Mexico, and from commercial sources. And that's what we see here. So in green, you have the most virulent nematode from Mexico, in blue, the most virulent from Rwanda, and in purple, the most virulent from commercial sources. And we tested those 5 nematodes against second instar caterpillar, third instar, and 6th instar fall armyworm caterpillars. And we tested them at 3 different doses. And actually, all of these doses of nematodes, so 5, 25, and 125, are actually very low with regard to nematodes. We can easily go to up to 1,000 nematodes per caterpillar per insect. So this is all very low. And the first thing that we can see is that nematodes are highly pathogenic to fall armyworm at already low doses. So for example, 25 nematode can kill close to 100% of the caterpillars. So that's very good. So fall armyworm is particularly susceptible to nematodes. The second thing that we can see here is that actually local nematode can be as effective as commercial nematodes. That's what we can see in all of these plots. So that's also very good. So we can use local nematodes. We don't need to use commercially available ones. And then finally, there's no evidence of any advantage in using nematode from the area of origin of fall armyworm as opposed to nematodes that are coming from the invasive range. And this is what we can see when you compare the green and the blue bar. There is no particular trend. So now we can also see that we have a very good candidate, which is Sternernema carpocapsae from Rwanda. So it's the most effective strain from Rwanda that we had. But can we now conclude that Sternernema carpocapsae should be the nematode to use in general? Unfortunately, it's not that simple because the virulence of the nematode varies very importantly between strains within the same species. And that's what we can see on this example where we have nematode from Mexico. So all of these nematodes were isolated in Mexico. And the colors on this plot represent different species. And for example, if we take that strain, which is Heterorhabditis bacteriophora, it is like 3 times more virulent than that strain, which is also an Heteroptelis bacteriophora. So it seems like if you want to use nematode for a biological control program, you need to do a screening of the virulence of your nematode candidates, because we cannot necessarily say that a species is more pathogenic than another. It's very strain dependent. Now, how to apply nematode above ground. So the easiest solution would be to simply spray them with water, but there is no evidence, the evidence suggests that nematode applied in water does not provide sufficient Fallet control, and this is evidence from the literature. So it seems like water is not good, and the reason to explain that is that water, when you apply it in the well, it will leak out, so seep out of the well, and then the nematode will desiccate and die. So with that in mind, we decided to incorporate nematode into different formulation that will remain onto the plants. So maybe using something that is more viscous, that will stay on the plant and plants and not leak out, or that could also increase the protection. So for this, we then tested 2 different types of gels, 2 different types of surfactants that are commercially available. We also use sand, as we heard in the previous talks, and like farmers apply sand in the well of a maize plant to control fall armyworm. This is particularly used in Africa, or at least it was used in the early step of the invasion. So we thought that by incorporating nematode in the sand, that will increase the efficacy of this technique. And we also tested some beads and some capsules. So now I will just show you a little video that introduce a little bit these formulations and the project. So here we can see some beads that we can produce in the lab. Then we can put nematode inside of these beads. We can also put maize juice in the beads, and then the fall armyworm is happy to feed on that bead, as we can see here. And once the fall armyworm ingests the nematode, it will die, usually within 24 hours. And then after a couple of days, the nematode will reproduce inside of this cadaver, and they will eventually come out in a very large number. And this is what we can see here, plenty of nematodes coming out of this cadaver. Then after some preliminary experiment with these different formulations, we found out that unfortunately the sand and the beads that I just showed were not giving so promising results, most likely because they are all drying very quickly, and then the nematode dies. So this is a bit the same problem than with water. So we decided to stick to 1 gel and to 1 surfactant, which I will from now on call SPF for surfactant polymer formulation. So this is a commercially available product. And we also included water because it's the easiest and the cheapest way of applying nematode, even though the literature suggests that it doesn't work so well. So it's a little bit of a negative control here. So we tested this formulation in laboratory trials first, where we had cages in which we placed 2 plants. And then we artificially infested this plant with 3 third instar fall armyworm caterpillars that we placed into the well. And then we let the insects establish on the plant for 24 hours. They started to feed already. And after this 24 hours, we applied our treatments into the well. And then 6 days later, we evaluated plant damage and caterpillar survival. Here is an example of what we could see after 6 days. So on the left, you have a plant that was infested with fall armyworm and then treated with water only without nematode. So this is a control. And we can see if you look at the well, don't know if you see my mouse, but the plant is quite severely damaged. And on the right, you have a plant that was treated with gel containing nematode. And you can see that the well is now intact, which means that the insects are probably dead. So for the damage, we used the Davis scale to record damage, where a 0 represents no damage and a 9 represents a plant that is almost totally destroyed. So basically the purple, blue are low damage, and the orange to red are severe damage. And on the vertical axis, you have the proportion of each of these damage scores that are attributed to the different treatments. So the first 3 treatments are the formulation without nematode, so this is control. And then the 3 last bars are the the formulation with the nematodes. So this is the treatment. And if you look at the pattern, there is a lot of red in the formulation alone, and then a lot of blue in the nematode treatments. So it indicates that plant damage is considerably reduced by nematode treatment, with intermediate efficacy for water with nematode, as expected. Regarding caterpillar survival, which we have here on the vertical axis, we have again the tree formulation without nematode first, and then the tree formulation with nematode. And we can see again that the caterpillar survival is considerably reduced by nematode treatments, with intermediate efficacy with water and extremely good results with the gel, where we had 0% survival. So this is very encouraging, but of course, this is laboratory experiments, so the conditions are much better than realistic field conditions. So then we brought these formulations to Rwanda, where together with a team of Rwandan scientists, we tested these into realistic farming condition with fields like that. We had 4 fields like this one, and we divided these fields into plots, that then each of these plots were treated with one of our treatments. And in this set of experiment, we applied our treatment only once, so it's one application, and we assessed plant damage and fall armyworm infestation 5 days and 10 days post-application. There is an example of how we applied our treatment. So for the liquid formulation, which were nematode in water, nematode in the surfactant, and also pesticide that we used as positive control, we used little hand sprayer, but we could also we could have used conventional spraying equipments that farmers have to spray larger areas. And for the gel, we use small syringes because they are very precise, and we can apply consistent amount of gels on each plant. But here again, we can use bigger devices such as glue guns, such as the one that you would use to apply silicone. And then this device could allow you to treat much bigger areas. Then after a couple of days, we were very happy to see that we found dead caterpillars on nematode-treated plants. So this was an indication that the nematode could also control fall armyworm under field conditions. So we are quite happy. And for the results, again, this is damage with the Davis score, where blues are little damage, orange, red, very severe damage. We have the result after 5 days on the left and 10 days on the right. And on the bottom, we have the different treatment that we used. So first we have a water treatment, which is simply a control. We just sprayed water on those plants without nematodes. Then in the middle, we have water, SPF and gel plus SC. So this is the same formulation that I showed you with the laboratory results. And on the right, we have cypermethrin, which is a pesticide that we use as positive control. And the second bar is actually water plus HR. So this is a second species of nematode that we applied in water just to compare the efficacy of these 2 different species. So what you can see here, if you look at the general pattern, it's not when you look at the general pattern, basically you see that the red, orange areas are decreasing towards the nematode treatments. So it indicates that nematode treatment reduces plant damage, and it does so to a level that is similar to what was achieved with the pesticide. And this is true at both 5 and 10 days post-application. Regarding caterpillar survival, so the level of infestation in our plots, we have caterpillars, the number of caterpillars that we recorded on the vertical axis in a total of 10 plants. And we again see that the number of caterpillar decreases with nematode treatment. And again, that the nematode treatment can be as effective as the pesticide cypermethrin to control fall. So I would agree with you that this is only a 40% reduction of infestation, which is not extremely impressive. But what is pretty nice is to see that we achieved a similar control than what was achieved by the pesticide. And maybe one explanation for only a 40% reduction is that our field were extremely heavily infested by fall armyworm, and maybe in region where there will be a little bit less pest, it will be more impressive. But it's already quite nice to see that we have an efficacy that is similar to pesticide. Then we did a second set of field trials, but this time with repeated application. So we went again to Rwanda. We had fields that we again divided into small plots that were each treated with a different treatment. And we this time applied our treatment as soon as we observed caterpillars, and we then reapplied the treatments every 2 weeks, so at 2 weeks interval until the flowers emerged. So we had 6 fields, and 4 of these fields were treated 4 times because the pests arrived at the very beginning of the season. So basically the first treatment was applied at this stage that you can see now on the picture. And in 2 other fields, the caterpillars arrived a little bit later. So we could only apply 3 treatments, 3 applications of the treatments. And so it started basically 2 weeks later. And then we assessed damage 7 and 14 days after each application of the treatments. And we also evaluated fall armyworm infestation 5 days post the 3rd application. So we did this only once through the season. And finally, we evaluated the yield. So here are the results for the damage. Again, we use the daily score, blues, little damage, and orange, red, severe damage. And this is the damage that are pooled for all fields and all applications. And what we can see is the result after 7 days and 14 days here. And we can see that, again, the nematode treatment can reduce plant damage to a level similar to the pesticide. And what we can see also is that we have a rather strong effect at 7 days, but it's a little bit minimized at 14 days. And that could mean that basically nematode treatment and also pesticide in this specific case may need to be applied weekly and not bi-weekly. So we had a good effect after 7 days, but not so much after 14 days. So potentially we should apply every 7 days and not every 14 days. Then, as I said, we looked at fall armyworm infestation levels. So this is how the plant looked like after the 3rd application. Then we destroyed some of these plants to look for caterpillars. And what we saw here on the vertical axis, you have the number of caterpillars per plots, which we sampled only 10 plants. And we can see is that we have a strong reduction of the number of caterpillars in the gel treatment as opposed to the control. This reduction is of approximately 50%. So we have 50% fewer caterpillar in the gel. And you probably also noticed that the pesticide treatment was not that effective here. And the reasons for that, so we have 2 reasons in mind. The first one is the rain. So after the 3rd application of our treatments, in 4 out of 6 fields, we had like very strong rains in the evening, and it's possible that the pesticide was washed away, whereas the gel treatment, which is way more viscous, will not be affected as much by rain. And the other possibility and possible explanation is that we used a suboptimal volume. So basically from the first application to the last, we always applied the same volume of treatments that we applied in the well. So for the liquid formulation, it's a spot spray. And for the gel, we applied it with syringes. And so basically when plants were small, this is sort of like a lot of treatments for a small plant. But later on in the season, when we applied, I don't know if you see my mouse, but the pesticide as a spray into the top leaves where fall armyworm are mostly feeding, then there is not much of this pesticide that will reach the lower leaves, and then the pesticide could control the insect feeding on the top leaf, but maybe less so on the bottom leaves. But what is very interesting as well here is that that the gel seems to be still working, even though it has the same application. So it was the same volume of gel. And it indicates that potentially the nematodes, when you apply them on top of the plants, they can then actively look for insects that are located elsewhere on the plants. So that's quite nice, even though our comparison with pesticide is not very easy to do with these results. Then regarding the yield, on the vertical axis, you have the yield expressed as ton per hectare of fresh weight maize. And we can see that we have basically 1 extra ton produced in the per hectare in the gel treatment. So this is a 16% increase in yield. So that's pretty encouraging. But again, it's difficult to compare the efficacy of the gel with the efficacy of the pesticide for the reason that I just mentioned, which are that maybe the volume applied was a little bit low. But anyway, we are quite happy to have seen that like we increased yield by 16%. So I hope that with this, I convince you that nematodes have great potential against foe. So obviously, at this stage, we don't have a ready to apply solution and a ready to apply to produce products. But I hope that like this is good proof of principle that convince you to look for more stuff with nematode and fall armyworm. Good thing with nematode is that the local ones were as effective as commercial ones, and they can be produced locally without relying on external sources. So for example, countries could produce their own nematode without relying on external sources, and then develop their own biopesticide. It seems that to be truly effective, nematode need to be incorporated in special formulations, such as in a gel, as opposed to simple water application. So we saw that water application had only intermediate efficacy. And we also saw that frequent application, for example, weekly application may be needed to achieve high efficacy, or at least in our case, with very high infestation levels, a biweekly application was probably not enough. And finally, we believe that the addition of apparent protectants and or fall armyworm attractants in the gel may increase its efficacy. So for example, it would be possible to add UV protectant or anti-desiccant in the gel to increase the longevity of the nematode and then their efficacy. And also, as we saw in the video, we could put fall armyworm attractants in the gel or in the bead here, and then the fall armyworm will potentially feed on the substrate rather than just passing by, and then this will also increase the efficacy. So with this, I would like to thank the funding for supporting our work, and also all the people that contributed to this project. And of course, I would like to thank you for your attention. Thank you very much. That's it.
Thank you very much, Patrick. Very interesting presentation. I need to clarify something here, aside from your talk. So our schedule here is a little odd, but we have a break scheduled for now, and then the general discussion. But if you can, I'd like you to stay in case there are any questions, and maybe we can take them now, or maybe we can all adjourn for a few minutes and come back to the general discussion. I'd rather ask you some questions now, and people may leave and come back for the general discussion at 14. Geez. I'm looking at our schedule and it's a bit off, but anyway, let me say this in Spanish now. Lo que sigue es una discusión general entre los ponentes que estamos por aquí todavía, y ustedes, el público. El tiempo nos hemos retrasado un poco, entonces le he pedido a Patrick que se quede con nosotros y le hacemos preguntas a él. Ya que se agoten estas, ya abrimos la discusión general. Entonces, los que gusten tomar un break por unos 5 minutos y regresar, aquí vamos a estar todavía. Okay? So I'll begin with a question for you, Patrick. I really like your presentation, very encouraging results. I mean, I'd been following your publications already, and actually used, I think 2 of them in class already. So fresh off from the journal, they were used in class. So I know you said this is proof of principle and it looks very good. Has anybody, obviously not you, you're too busy with your own thing, but somebody in the engineering areas, has somebody begun looking in how you would apply gels in maize fields? Let me repeat this question in Spanish. Estoy preguntando a Patrick que si alguien en las áreas de ingeniería se ha empezado a ver la manera de cómo se aplican geles en cultivos de maíz, ¿no? Porque supongo que no es algo fácil. Okay, all yours, Patrick.
Yeah, thanks for your very nice comments and for your question. So yes, as you guessed, I didn't look into myself like specific machinery to apply. We used so in the presentation I showed syringes, but like big glue guns also work, but it's obviously all manual. But like for larger scale farming system, there is actually a company that is close, not too far from us geographically, which Ted approached. So Ted is my supervisor, Ted Turnings, and they discussed together. And then this company is actually making high-tech machineries for agriculture with automated robots that go through fields. And then they have like 3D cameras where they can actually they develop these robots to apply herbicide on weeds. So with the 3D camera, they could identify the weeds and then specifically apply the herbicide onto the weeds only. And then they discussed and they said that probably, but it would need to be tested, they could target the maize well and then apply the gel right into there. So further discussions are supposed to be coming. The problem is obviously we cannot, like they have their machine in Europe and we obviously cannot test our armyworm in Europe because the armyworm is not present here, but just to test whether their technique could work should be something that we test in the future, or my successor.
Very interesting. Nos comenta Patrick que ya hay una compañía cercana de allí de la universidad donde él trabaja, de ingeniería, que ya tiene robots para aplicar herbicidas de manera muy específica y que estos robots fácilmente se pudieran modificar para que aplicasen gel precisamente en el cogollo del maíz. Entonces esa es una posibilidad para el futuro. Any other questions out there? So the product from Cooper, could that be tweaked to make it a little better? Because it wasn't very different from the gel, right?
So the
SPF.
I didn't. Oh yeah, maybe you want to translate that in Spanish first.
Yes, preguntaba sobre el producto que vende Cooper, que es un no recuerdo cuál es la palabra, pero es para aplicaciones de microbiales y dio resultados no iguales al gel, pero similares. Entonces le pregunto a Patrick que si han pensado quizás empezar a modificar, a mejorar este producto que ya existe.
Yeah, so the first thing in the field trials, we used not the product from Coppert, but the product from Inema. So I showed at the beginning that we had two different surfactants. One was from Coppert and the other one was from Inema. And after preliminary trials, we had result with the product from Inema. So we tested this one in field conditions because obviously, unfortunately, we couldn't test all the formulation that we screened at the beginning. And I'm a little bit skeptical with this liquid formulation because yeah, like they seem to work rather nicely in the laboratory, but in the field condition, particularly in the second experiment, if you look at the number of caterpillar that we recovered in our plots, the gel was much better, and actually the liquid formulation was like rather intermediate in terms of result. And I think the reason for that is that you need to sort of fill up the well, in vegetative maize at least, to reach the caterpillar, because the caterpillars are not completely at the bottom of the well. They are slightly higher up, like a few centimeters, or not even few centimeters, 1 centimeter above the deepest point of the well, sort of. And this liquid formulation, even though they are surfactants, so they stick to the leaf, like most of it will seep down the well, whereas the gel fills it up completely, and then the caterpillar actually walk on top of the gels. I don't know really, but it seems that like the gel is still a little bit more efficient, but other formulation could also come to the market and also show promising results.
Sí, entonces la respuesta corta, básicamente yo aprecié mal el producto. Por un lado no es de Copper, es de otra compañía. Por otro lado, no funcionó tan bien, funcionó bastante bien en laboratorio, pero no en campo. Entonces aprecié mal ese punto. Y entonces hay la duda realmente de que si vale seguir intentando con el producto este, siendo que el gel funcionó tan bien. There's some questions on the chat here, and it has to do with relative humidity and whether this approach would be applicable in areas where humidity is low.
So yes, obviously the humidity is a very important factor. And in our case, we tested this in a tropical country where the humidity, there is always a little bit of rain through the day, particularly in the evenings. But when the condition will be very dry, I guess that's when the gels comes in very handy, because the gel is actually holding humidity because it's a gel. So then it provides the humidity to the nematode, or otherwise formulation will need to be developed with anti-desiccants. But definitely if the plant is completely dry, that's a problem. But also, I don't know how it is in very arid areas, but there is often always almost a little bit of water, like even coming from the dew and like the air humidity into the well where the nematode should be applied. And then this potentially could be just enough for the nematode to survive.
Sí, evidentemente los experimentos de campo se hicieron en un país tropical, Ruanda, pero también tenemos que tomar en cuenta que precisamente la función del gel es mantener la humedad que necesitan los nemátodos. También hace la observación, Patrick, de que aún en zonas muy áridas tienden a acumular humedad. Las plantas, sobre todo en el cogollo, no sé si seguramente se han fijado por ahí, y esto seguramente también ayudará al gel. Another question is, how specific are the nematodes, and do you have strains or biotypes that are more aggressive than others?
So yeah, nematodes are very generalist biocontrol agents, so they can attack a wide variety of insects. So definitely generalist biocontrol agents. And as I said, like some strains are way more virulent to a specific insect than other. And then if you change the insect, then it probably changes also the virulence of the strains. So yes, they are very generalist, and different strains have different efficacy.
Sí, entonces la respuesta a la pregunta de qué tan específicos son los nemátodos, nos dice Patrick que a nivel de especies son muy generalistas en cuanto a sus hospederos, pero que sí hay razas o cepas que son mucho más virulentas que otras. Entonces las diferencias están en cuestión no de especificidad del hospedero, sino en su virulencia. Y la virulencia puede ser más hacia un hospedero y menos hacia otra, y luego en otra especie puede ser lo inverso, ¿no? Entonces ahí es donde está la variabilidad. I'll add one more question before we turn to the general discussion real quick. So for the beads, the caterpillars were actually feeding on the beads, right? And you might even add some maize leaf flavoring to that. How did this operate with the gel? Are the nematodes leaving the gel on their own looking for a host, or are the caterpillars consuming the gel? How was that?
So actually, we just tested whether like what would be the effect of adding maize juice into the beads. We also did that into the gel. Like when I showed the formulation, actually caterpillars were feeding on the gel because there was maize juice inside, but we didn't include maize juice in any of the experiments. So the gel were like nature, so they didn't have any flavor that would attract the caterpillars. The reason for this is that getting maize juice, you need a lot of maize to do that. So it's not very sustainable, but we still have in mind to actually identify specific compounds that could be produced and then incorporated, and not maize juice. So maize juice, you need way too much plants for that. But just for the proof of principle, again, we looked at this and really, like, you can actually potentially make them feed on the substrate and not only just pass by, and then the nematode can jump.
I see.
Thank you for the question.
Me disculpo, no hice la pregunta en español primero, pero la pregunta fue de que si el caso del gel fue gel solamente o hubo que agregarle algún saborizante para que lo consumieran las larvas, y me dice que no fue así, que sí han probado agregarle saborizante y sí lo consumen las larvas, pero en los experimentos de campo fue solo el gel nada más, pero que ya empiezan a buscar compuestos presentes en el maíz que pudieran estimular el apetito ahora sí de las larvas, ¿no? Para que sea atractivo, para que consuman. Okay.